everyone. So we're going to be talking about the structure of DNA today. So hopefully you tuned in to the history of DNA. And so now we're going to be on part 2. We need to talk about all the stuff that, Rosalind Franklin and James Watson discovered with that shape of DNA. And we also need to know how to label it. So let's go ahead and get started. So I'm going to go ahead and open up my notes in my interactive notebook. And I'm going to go ahead and move to slide number 7. So these notes are extremely short. This is not going to be a very long video at all. So first off, we need to talk about what DNA actually is. So DNA stands for deoxyribonucleic acid, so DNA. So deoxyribonucleic acid we know is a polymer. Hopefully you remember that from when we talked about biomolecules way back in the day. So because this is a polymer, that means that we have smaller subunits to make up this molecule. And those smaller subunits are called nucleotides. So nucleotides are my monomers. So when I'm talking about a nucleotide, it's going to be repeating structures of 3. And so if I look right here, I have this little shape here with ABC. It's a little pentagon, a circle, and a rectangle. This right here is a nucleotide. And so I'm going to have repeating nucleotides in order to make up my DNA structure. So I have these little pink boxes over here at the side, and I'm just going to click and drag so I can label my nucleotide. So my circle is a phosphate group. My rectangle is a nitrogenous base. And my pentagon is a 5-carbon sugar. And in this case, the 5-carbon sugar is deoxyribose. When we talk about RNA, we'll talk about the other type of sugar that we could have, which is ribose. But for right now, let's just focus on DNA. So my nitrogenous bases are extremely important because they're going to code for my genetic traits. So we all have different sequences of these nitrogenous bases. And because we have different sequences, that means that we each code for different traits. So you received your nitrogenous bases from your parents, they received it from their parents, and so on. And so whatever nitrogenous bases you have, that's going to code for how you look, how you act, maybe even how you age. So all of that is going to tie into our genetics unit when we learn in a couple of weeks. So next, we need to talk about DNA. So DNA actually runs in opposite directions. Okay, so what that means is that one strand is going to be going maybe to the right and the other one's going to go to the left. So I may be mirrored on my video here. Okay, so maybe one's going to go up and one's going to go down. And so we refer to that as anti-parallel. And you'll see what I mean whenever I go to my next slide. So anti-parallel is when DNA runs in the opposite direction. We also know that bases are going to match. So when they match, we refer to those as complementary bases. So you may notice complementary is spelled differently than a complement. So complement would be like, oh, you look really nice today. I like what you did to your hair. Complementary with an E is going to be bases that are matching, almost like a puzzle piece. So hopefully you remember from our history of DNA, we have 4 bases for DNA. We have adenine, which we use the letter A, and adenine pairs up with thymine. We also have cytosine, which pairs up with guanine. So apples in the tree, cars in the garage. Okay, so that's what we use in order to make sure that our bases are matching. So our next slide, we're going to continue to label our DNA strand. Now this is a DNA strand that has been flattened. So it would be as if I took my strand of DNA and just completely flattened it. This is a long strand of DNA. Okay, so I would be looking, okay, at my ladder untwisted. So I can see on my ladder, one, I have bases in the middle. You can see all these bright colors in the middle. And on the outside, I have alternating white and black sections. This outside part is called a backbone. So my backbone are where my sugar and phosphate groups are located. The bases are all on the inside. So let's go ahead and label this DNA structure. So first off, if I look up here at this top box, I can see I have three shapes here. I have my rectangle, my pentagon, and my circle. I know from our previous slide that that is a nucleotide. Next, I can see I have lines that attach my bases in the middle. So all of my bases need to be able to connect, and they do so with a hydrogen bond. Now, hydrogen bonds are really important to know about for biology just because they allow reactions to occur that we need whenever we're making molecules. We'll talk more about that one later. So next, I'm going to go down here at the bottom and I can see I have a circle and I also have my pentagon. Hopefully you remember from the previous slide, my little circle is my phosphate group. My little pentagon is going to be deoxyribose, which is my 5-carbon sugar. So you can see for my backbone, I have alternating phosphate and deoxyribose sections, and I have one on each side. So remember how I told you that these strands run anti-parallel? Well, let's look at where those phosphate groups are pointing. So at the bottom, I could see my phosphate groups are all going to the left. Okay, so it's my left, probably, I don't know, I guess this is mirrored, so you're right. Okay, and then at the top, they're going the other way. So because they're going in opposite directions, that means that it's anti-parallel. So we're looking at the direction that the phosphate groups and the sugars are facing. And if we put letters here for our bases, so our A, T, G, and C, so the letters on one side would be upside down. So lastly, we have two more words here. We have pyrimidine and purines. So I'm actually going to skip back to slide number 5. And I have my nitrogenous base structures here. So I can see adenine and guanine. These are my darker gray rings, and there's 2 rings there. Thymine and cytosine, those are light gray rings, and there's only 1 ring there. Remember, in the middle, I have to have 3 rings total. So my darker gray rings are called purines, okay? And those are purines because they have 2 rings there. The ones that are lighter gray are my pyrimidines. They only have 1 ring there. So that means that I always have to have a purine pairing with the pyrimidine. So let's go back over here. I can see right here I have a little box. Okay, so because it's a little box, I know that it's my smaller structure, which is my pyrimidine. Right here I have my bigger box, so I know that that's a purine. I could also go a step further by looking at my hydrogen bonds. So my hydrogen bonds are what connects the bases together, and I can see right here I have 3 hydrogen bonds. Well, if I go back to slide number 5, I can see between adenine and thymine, there's 2 hydrogen bonds, and between guanine and cytosine, I have 3 hydrogen bonds. So I know that first set of bases is going to be guanine and cytosine. And then if I look back at that, I can see that I have the purine down at the bottom and my pyridine at the top. So I know that guanine is a purine and has 3 hydrogen bonds. So I would know that this box right here is guanine and it's going to attach to cytosine. Now, if I look at the second box, I can see that there's only 2 hydrogen bonds right here. So I know this has to be an adenine-thymine pair. But this one at the bottom is small. It's a pyrimidine. So let's pop on over and see which one is the pyrimidine. So thymine is my pyrimidine. So I know over here, thymine is on the bottom and adenine is at the top. So for my next one right here, I can see it's the same thing. I have a small box on the bottom, a big one at the top, and then 2 hydrogen bonds in between. So again, I know that this one right here is thymine and this one up here is adenine. So I could keep on going if I wanted to, but I've gone ahead and labeled everything that I need to. So hopefully you understand this. If you have any questions, let me know. Those of you in my class, go ahead and hop onto your Schoology page. That way you can do your Ed puzzle. Bye.